Hello students, this is Vedant Mirza. We are discussing the respiratory system today. Respiratory system involves respiration and breathing. Respiration is about exchange of gases which occurs between the atmosphere, blood and cell. It is a combination of three processes: ventilation, which involves breathing; external pulmonary respiration, where the gas exchange occurs at the level of lungs; and internal tissue respiration, where the exchange occurs at the level of tissue. The cardiovascular system assists the respiratory system by distributing the gases and taking them to the actual cells of the body. The structures of respiratory system include the upper respiratory system and the lower one, and functionally they include the conducting zone and the respiratory zone. The conducting zone is where a gas exchange does not occur, and respiratory zone is where the exchange occurs. So the structure of respiratory system involves your nose, your pharynx, your larynx, your trachea. Here, the way that the position at which the trachea they split into two bronchi. This point is called carina. So the right bronchus and the left bronchus they move to the individual lungs, and you can see that outside the lungs there is enclosed in the rib cage, and in front you have sternum. So internally, this is how your lungs and your rib cage actually looks like. If you have cut the ribs open, this is where the thoracic cavity is. The respiratory system upper include the nose, pharynx, and the associated structure. The lower system include the larynx, trachea, bronchi, and lungs. The overview of if you go to the internal anatomy, you can see that there are empty spaces in your facial region, which are called as the sinuses. These sinuses are surrounding your nose. There is olfactory epithelium present in your nose, through which you can sense the different smells in your surrounding. The nasal conchae are these regions within the nose. These they have a large surface area, so when the air goes into your external nares are the nostrils. When it goes here, it strikes with these nasal conchae. The air, its velocity decreases. It basically gets warmed up as well, and from the nasal conchae it then moves on to this region, and from here it goes into the trachea. So you can see that the region in your throat, this includes various structures. This includes your pharynx. The pharynx includes the oropharynx. Initially you have the nasopharynx, then you have the oropharynx, then you have laryngopharynx. These three parts of pharynx, and then you have your the trachea begins, and then in your larynx there are various cartilages. There you have thyroid cartilage, the cricoid cartilage, and the thyroid gland present here in these regions. The trachea is the tube that will take this air to your lungs. Then your nails actually is made up of cartilages. These are the alar cartilages. These included they are present in your lungs. They lay along in your nose, sorry, not lungs, in your nose. And this is the septal cartilage at the center. Then there are two lateral nasal cartilage. These are just cartilaginous structure. There is a dense fibrous connective adipose tissue at the end of here. This is the bony framework of your nose, the frontal bone, the nasal bones, and the maxilla. The internal anatomy, as I've already discussed, include nasal conchae and the associated structures. The nasal conchae, they allow the air to comes into contact, and the air loses speed, and it gets warmed up as well. In doing so, it gets filtered as well. So the surface anatomy of nose include the root, the apex, the bridge, and the external layers. You can see this is the root. This is the apex of the nose. These are the external layers, which you call as nostrils. This is the bridge. Here you will have internal nares. Internal nares means they're the internal roots, internal these nostrils. These are the external nostrils, and in between there is a cartilage that separates the two. Pharynx, as I told you, is divided into three parts: nasopharynx, oropharynx, and laryngopharynx. This is the nasopharynx, the oropharynx, and the laryngopharynx. This basically actually tells you that this part of pharynx is near the nose, this oropharynx is near the anatomically near the oral region, and the laryngopharynx is close to the larynx. The larynx is your voice box. It basically connects your pharynx and the trachea. The larynx is basically is having various parts, and when they vibrate in contact with each other, so voice is produced. So what you have epiglottis at the top, then you have a hyoid bone. These are the thyrohyoid membranes. Then the epiglottis leaf is there. This is the epiglottis leaf. This is the stem of that leaf. Then you have corniculate cartilage. You have thyroid cartilage, the Adam's apple here, which you can see in the neck of many people. Then you have arytenoid cartilage. These are arytenoid cartilage. Then you have cricothyroid ligament. This is the cricothyroid ligament. Then you have cricoid cartilage here, and the thyroid glands and the parathyroid gland. These are the four parathyroid glands that we have discussed in our endocrine system. And then the tracheal cartilage begins. Larynx contain the vocal fold, which basically, when vibrate, they produce sound. The structure of voice production, this include the thyroid cartilage, the cricoid cartilage, the vocal ligaments, and the arytenoid cartilage. The posterior cricoarytenoid muscles is there as well. And when you are producing the sound, the either this opening either it just this opens up or this closes. So it depends that when viewed, it either the vocal folds they move together, either they are abducted, they are moved apart, or they are adducted. Adducted means that they are close together. So these are the anatomical term, abduction and adduction. The vocal folds, these are the vocal folds in this larynx region, and this region here is called as the rima glottis. These are the vestibular folds, and these are more cartilages. You don't really need to learn all the name of these cartilages. Trachea. Then trachea, you can see this trachea and esophagus when cut in section. You see that trachea is present in the front part of the region. This is the sternum here. This is the trachea, and then there will be a trachealis muscle here, and then there will be your esophagus at its back. So bronchi, the superior border is actually at the fifth thoracic vertebra, and then trachea just branches off into the left and right bronchi. This is the point of location of carina that I told you. You can see that the bronchus is going there. The bronchus then divides into right main bronchus. The right main bronchus will then further divide into right lobar bronchus. Lobar means each will go into each lobe. So right lobar bronchus, then there are segmental bronchus. Segmental means that is further divided, and then the bronchiole that starts to form. The bronchioles will keep on dividing till they form the terminal bronchiole. The same thing goes on the left hand, on the left side as well. But since the left region of your lungs is not having the same number of lobes, the number of lobar bronchus will also differ between right and left lung. This is the pathway how they just basically occur. The terminal bronchioles are the end of the conducting zone, and at the end of this all was the conducting zone. No gas exchange occurs here. But when you go to the conducting zone, this is your alveolar duct and the alveoli. This is where the exchange occurs. So the lungs, they are enclosed in the pleural membrane. The pleural membrane, it is having pleural fluid in between. When you were discussing the lungs, the lateral view of right lung and the lateral view of left lung, you can see that the right lung is having an apex. They both have apex. They both have superior lobe. The horizontal fissure is there, and the horizontal fissure is there together with the oblique fissure. And how many lobes do you see? You see an inferior lobe, you see a middle lobe, and you see a superior lobe. So in right lung, there are three lobes. In the left lung, there is just an oblique fissure which separates the inferior lobe from the superior lobe. There is no horizontal fissure here. There is no middle lobe here, because the left lung and right lung, if you just compare, the left lung is a bit smaller because it needs to have a cardiac notch. In the cardiac notch, heart is present. So when you come to alveoli, alveoli is actually the respiratory zone where you have air sacs. So this is the alveolar duct. These are the bunch of alveoli. These alveoli have these are all are alveoli. This entirely is an alveolar sac. This will have a capillary network around itself, and all these are single cell linings. The alveolar duct is also a single cell lining. So combinedly, this is the region where gas exchange occurs. The type of cells inside the alveoli, these include the type 1 and type 2 cells. The type 2 cells of alveolar, they are called septal cells. So these are the septal cells. This is all single cell lining. Then type 1 cells are also shown to you here. These are the type 1 cells. Alveolar macrophages are also shown. These are the ones that will phagocytize any pathogen or any dust particles that come here. These are the red blood vessels. The exchange occurs through this region. This is the respiratory membrane. The respiratory membrane, it includes how the capillary endothelium, the capillary basement membrane, the epithelial basement membrane, they are involved. Type 1 alveolar cell is the alveolar cell which is involved in the exchange of gases here. Respiratory membrane, as I told you, they include these four layers: a layer of type 1 and type 2 cells, an epithelial basement membrane, a capillary basement membrane, and the capillary endothelium. This is how the exchange can occur. And once the gas exchange, it goes down the gradient from high concentration to low concentration. The blood enters from the pulmonary circulation, which actually was a branching off of the systemic circulation, and the blood exits by the pulmonary veins. The ventilation perfusion coupling occurs, and blood gets oxygenated. This is a summary of the you can say how the cells and the description differs. So in the nose region, you can see with the epithelium is non-keratinized, pseudostratified, and olfactory epithelium having olfactory receptors. They have cilia, but the non-keratinized do not have cilia. Goblet cells are not present in all except for the pseudostratified ciliated one. they contain the goblet cells which produce mucus. When you come to pharynx, the naso oral and laryngopharynx, they are of pseudo-stratified ciliated columnar, non-keratinized stratified squamous, and non-keratinized stratified squamous. So they both are of the same kind of cell. They are a bit different. They pseudo-stratified the nasopharynx region is having cilia, it is having goblet cells, but the oropharynx and the laryngopharynx, neither of them have cilia, neither of them is having goblet cells or mucus production ability either. If you come to larynx, it is non-keratinized stratified squamous cells above the vocal folds, and after that they are pseudo-stratified ciliated below the vocal folds. No cilia present above it, but yes, below it it's present. No goblet cell present above it, but yes, below it is present. So the trachea, it's basically made up of pseudo-stratified ciliated columnar cells. Yes, cilia is present. Yes, goblet cells are present. This cartilage is C-shaped. If you just discuss the bronchi, you see they are made of pseudo-stratified ciliated columnar epithelial cells. The cilia is present throughout. Goblet cells are present throughout except for the smaller bronchioles, where the goblet cells are not present. In terminal bronchioles, neither cilia nor goblet cells are present. In lungs, when you enter the lungs, the respiratory bronchiole, the alveolar ducts, and the alveoli, which these three respiratory structures, you don't see cilia or goblet cell in any of these. Pulmonary ventilation is about inhalation and exhalation. How because of pressure changes, the respiratory muscles actually bring about those pressure changes, and air flows in, and it follows Boyle's law. Boyle's law is about how the volume of gas varies inversely with its pressure. So when your muscles of your inhalation and exhalation they are involved, they change the volume of your thoracic cavity, which in turn changes the pressure and the air flows in. Which muscles are involved here? The main muscle is your diaphragm. This muscle is the major muscle of respiration. So when diaphragm is pushed down, diaphragm goes down, you can imagine that the thoracic cavity volume increases in this vertical dimension. And when the external intercostal muscle, the intercostal muscles, they get contracted, the rib cage get raised upwards and outwards, just like a handle of a bucket. So when it goes upwards and outwards, radially the rib cage goes up and away from your lungs. It increases the volume within the thoracic cavity, the diagonally as well as laterally as well as medially. So the volume increase here reduces the pressure within the thoracic cavity. This allows the air to go in. When the air goes in, this is how the pulmonary ventilation occurs. And for exhalation to occur, the diaphragm again contracts. The diaphragm relaxes. The diaphragm relaxes, it goes up. When it goes up, it pushes the lungs from the bottom. The external intercostal muscles relax as well. And the oblique muscle, the internal intercostal muscles are then involved in how exhalation occurs. So actually, inhalation is an active process. It involves the contraction of muscles. Exhalation in most of the time is passive. It does not involve any contraction, but when forceful exhalation needs to occur, that will be active as well. This is how the rib cage moves in the way that I told you that the bucket handle moves. And the other factors affecting pulmonary ventilation include surface tension. This is basically present in your alveoli because of the gases being there. So alveoli must overcome this to expand the lungs, and this is surface tension is lowered by the surfactant which is present in your alveoli. The cells have a moist lining. This lowers the surface tension. The elastic recoil decreases the size of alveoli during expiration. So elastic recoil basically just tell you the idea that when the alveoli will stretch, they can recoil back and gain their original shape. Compliance is just the ease with which the lungs can expand. These are some modified breathing movements which you can just go through: how coughing, sneezing, sighing, yawning, sobbing, crying, laughing, hiccuping, Valsalva maneuver, which you actually go on when you are passing out feces, or pressurizing the middle air. All these are the breathing movements that we modify and we learn them through our life. And these are just the description mentioned. This is something very good for information. When we are discussing lung volumes and capacities, we discuss some lung volume and capacities here. This graph actually is taken from an experiment which we performed using a respirometer. This is the tidal volume. This amount of gas is basically what we exchange in every breath. So this is about 500 ml. Every now and then we take a deep exhalation and a deep inhalation. forceful exhalation, and forceful inhalation. So here we have inspiratory reserve volume. This means additional air that we can breathe in upon forceful inhalation. Expiratory reserve volume is the additional air that you can push out upon forceful exhalation. The values are 1900 ml and 700 ml respectively. Then there's an air residual volume about 1100 ml, which you just cannot push out of your lungs, this no matter how forcefully you exhale. So this stays there. So these are the main four volumes that you have to keep in mind, and their definitions and their volumes as well, and how to label these on graph. Then we have capacities as well, which capacities are, for example, the total lung capacity. Total lung capacity is 4200 ml. This includes your entire all the volumes combined, the inspiratory reserve volume, the tidal volume, the expiratory reserve volume, the residual volume. Combine them together, this is the total lung capacity. Vital capacity is the air that you vitally exchange. You don't exchange the residual volume every now and then. So this much stays there. So you subtract total lung capacity, you subtract the inspiratory and residual volume and that expiratory reserve volume from total lung capacity, you will get the vital capacity. Your inspiratory capacity is inspiratory reserve volume together with the the tidal volume, and your functional residual capacity is your respiratory reserve volume combined with the residual volume. The exchange basically is governed by Dalton's law of partial pressure and Henry's law as well, which basically tells you respectively that each gas in the mix, if a mixture of gas exerts an more pressure as if no other gas was present, that's Dalton's law. So oxygen and carbon dioxide, they behave very independent of each other. Even nitrogen behaves very independent. Although there's not much influence of nitrogen when it comes to gas exchange, but yes, it's very good in understanding the actual respiratory physiology. Henry's law is the quantity of gas that will dissolve in the liquid is proportional to the partial pressure of the gas, and the solubility coefficient within the temperature remains constant. So during external respiration, what's happening is basically carbon dioxide and oxygen, they both move in opposite direction depending on the diffusion gradient. You know your lungs will have a high partial pressure of oxygen when you have just inhaled, and your deoxygenated blood will have a high volume or high partial pressure of carbon dioxide. So carbon dioxide will enter the alveolar space, and the oxygen will go from the alveolar space into the blood, and the blood will become oxygenated. When this goes to the tissue, at the level of tissue cells, the tissue cells will have a high PCO2, high partial pressure of carbon dioxide now, and your blood is oxygenated. It's having a high partial pressure of oxygen. So oxygen diffuses into the tissues, and the carbon dioxide diffuses into the blood in opposite directions down their diffusion gradients. This is exactly shown here to you in this table, in this you can say mind map. You can see how the exchange of CO2 goes into the alveoli, oxygen comes here into the pulmonary capillary, this goes to the left atrium. From here you can see the blood is distributed throughout the body through systemic capillaries. At the tissue level, oxygen diffuses into the cells, carbon dioxide comes back into the blood, the deoxygenated blood returns to the heart into the right atrium. It goes to the pulmonary circulation and it enters the lungs, and this is how this deoxygenated blood and oxygenated blood are continuously circulated, and external respiration keeps occurring between the pulmonary capillaries and the alveoli, and internal respiration keeps occurring between the systemic capillaries and your cells. The transport of oxygen basically is 1.5% is dissolved in plasma, and about 98.5% is carried in hemoglobin. When we come to carbon dioxide, the blood is playing a very important role in carbon dioxide transport as well. 7% is dissolved in plasma, and 23% is carried inside hemoglobin. In red blood cells as carb aminohemoglobin, where carbon dioxide binds to the amine group of hemoglobin. That's why we call it a carb aminohemoglobin. And 70% of CO2 is transported as bicarbonate ions. So there are various factors affecting the affinity of hemoglobin for oxygen. The black curve here actually shows you the normal hemoglobin, how the hemoglobin saturation actually is affected by the partial pressure of oxygen. The more oxygen that you have, the more saturated the hemoglobin will be. And this is an S-shaped curve. So there are cases in which this curve either shifts up or it shifts down. For example, if the blood pH is low, if blood pH is low, the graph would go up as shown by this blue graph. The same will happen with low PCO2, because you know low PCO2 means high blood pH, because CO2 itself is an acidic gas. When there's a high PCO2, it means the pH will drop down to an acidic level or below neutral, or it will be going low blood pH. So high blood PCO2 basically means low blood pH. So you can see the graph going here. So the shift is either above or either below, depending the whether the blood pH is high or blood pH is low. In this way, we actually understand the partial pressure of how much oxygen and how much your blood can stay saturated with oxygen. So for example, if your tissue is having just 30 millimeter mercury of oxygen at a level, and normally the blood, if it were the blood passing, it had a high blood pH, then it would still keep this much of oxygen saturated, it would leave this much of oxygen to the tissue. But if the blood had a low pH, so in that case the saturation would be different. The blood will end up losing this much amount of oxygen to the tissue. You can see how temperature affects this as well. So with the high temperature, this graph shifts down. At low temperature, it goes. Affinity for hemoglobin for oxygen in fetus is more than the maternal. That's why the fetus can take oxygen from the mother's blood supply. This diagram here it shows to you the Bohr's effect and the Haldane effects, how the blood actually picks up oxygen and how it delivers it to the cell and picks up carbon dioxide there. So what's happening at the exchange of oxygen at the pulmonary level at the capillaries here? You have oxygen and high level of oxygen in the inhaled air. It passes through the tissue fluid. It crosses the basement membrane of the capillary wall. It enters red blood cell. Inside the red blood cell, oxygen bind with hemoglobin. Hemoglobin till now was carrying H, the hydrogen, H plus ion with it. This was hemoglobinic acid. So oxygen combined with HBH, and how hemoglobin gets oxygenated, it loses this H plus ion. The H plus ion then binds with the bicarbonate ion and combines to form H2CO3. H2CO3 by the help of enzyme carbonic anhydrate dissociates into H2O and CO2. They both move out into the plasma, and while they are going out, since this is going out, at the same time, a chloride shift also occurs, reverse chloride shift occurs, chloride ions go out as well. So carbon dioxide, it goes out and it enters into the alveoli and is exhaled. This is the reverse exactly at the level of tissue where your blood was having oxygen. So here, what is happening, you had carbon dioxide at the level of tissue. The carbon dioxide from the tissue moved to the interstitial fluid. It crossed the systemic capillary, it entered into your red blood cell. Once inside the red blood cell, it allowed chloride ion to enter as well. When the chloride ion and carbon dioxide they both enter together, carbon dioxide combined with water, the carbonic anhydrase enzyme allows H2CO3 to form, which dissociates into HCO3 ion. HCO3 ion then goes out. When HCO3 ion actually going out, this is negative ion. This is where the chloride shift occurs. So bicarbonate ion leaving allow chloride ions to move. Here the bicarbonate ion was entering, so chloride ion had to move. So reverse chloride shift and forward chloride shift. So chloride shift actually occurs to balance out the bicarbonate ion. So chloride enters and bicarbonate leaves. The H plus is left here. So H plus actually was the proton that was formed from the combination of H2O and CO2. So this H plus it combined with hemoglobin and oxygen. The moment H plus bind with hemoglobin must let go of oxygen, so oxygen is released. So you can see that this reaction is exactly the reverse here. At the level of alveoli, when oxygen was coming and attaching itself to HBH, H would be lost and oxyhemoglobin would form. Here, oxyhemoglobin was there. When H plus attached to it, oxygen was released. So this is how internal respiration and external respiration differ from one another. Your control of respiration is basically occurring in your brain stem. There are respiratory centers, which are the pontine respiratory group. This is the respiratory center, the pontine respiratory center. This is a group of neurons. They control respiration. Then the medullary respiratory center include the prebronchial complex, the dorsal respiratory group, and the ventral respiratory group. They allow the nerve fibers to go through. The phrenic nerve is there, and this phrenic nerve it actually innervates your diaphragm and the muscles which are involved in respiration. So they control the contraction and relaxation of external intercostal muscle in the diaphragm. As I told you that when the signal will be passed from here, diaphragm will be contracted, external intercostal muscle will get contracted, and they will go. The diaphragm will be pushed down. The external intercostal muscle will be raised upwards and outwards. This would increase the volume within the thoracic cavity and the air would flow in. The control of respiration, this is given in this flow chart, how the dorsal respiratory group activate and inactivate within few seconds the diaphragm and how it can cause the contraction relaxation during normal quiet inhalation and exhalation, and how the dorsal respiratory group of neurons can control the ventral respiratory group of neurons, and how basically they are again allowing the muscles contraction during forceful inhalation and forceful exhalation. So this is under normal quiet breathing. This is under forceful inhalation and forceful exhalation. The control of respiration also involves cortical influence, which means you can take conscious activities. For example, you can just decide to hold on your breathing, and there are chemoreceptors in your peripheral your body. They constantly monitor amount of oxygen and carbon dioxide, and they send this input to your brain. Hypercapnia has a slight increase in PuCO2 and hence the H plus, where it stimulates the central chemoreceptor, and hypoxia is the deficiency of oxygen at the tissue level caused by low oxygen in the blood. This is a summary of the various stimuli that affect the rate and depth of breathing. This includes the hyperventilation, it includes arterial blood pressure, and how various body temperature and activities of proprioceptor, play pain, blood pressure, and stretching of the anal sphincter. All of these, they are the stimulus that decrease the rate and depth of breathing. Rate of what do you mean by rate of breathing? Rate of breathing means how many breaths are there in per minute, and depth of breathing mean how deep do you breathe? How shallow is your breathing? Either you could be taking shallow breaths or you could be taking deep breaths. Overall, the homeostasis is served by respiratory system. It contributes it at the level of muscular system, the nervous system, endocrine system, cardiovascular, reproductive, urinary, digestive, as well as the lymphatic system. So respiratory system, you can say, is governing almost all the organ systems of the body and maintaining homeostasis. This is it for now. Thank you so much.